Hello friends, this video on biomolecules part 27 is brought to you by examfear.com. No more fear from exam. Now if you see, the nucleic acids are used for DNA fingerprinting. So what is DNA fingerprinting? See, every individual has its own unique fingerprint, right? But the problem is this fingerprint can be altered by surgery. You can do a surgery and get your fingerprint changed. So we need something which cannot be changed, but this is a fingerprint. If you see, this is unique for a given person. That's why the thumb impression is valid as a signature. This thumb impression is valid as a signature. A lot of places it is used. If you go and try to get your passport done, they'll ask you for the thumb impression. But this can be altered with a surgery. So what is the best option? So this sequence of DNA which we see can also be used to identify a person, because this is unique for a given person. Also, till now there is no treatment which can change this DNA sequence. Correct? First, it is unique and it cannot be altered, so it has fair advantage over fingerprinting. Correct? So it can be used in forensic labs to identify criminals. It can be used to determine the paternity of an individual. You have a kid, you want to know who is the father, you can use this. It can also be used to identify the dead bodies in any accidents. If there is an accident and there are a lot of dead bodies, they want to know who the person is because the whole body is shattered, then the DNA can be used. It can also be used to understand the biological evolution. This DNA is used in a lot of places, right? So it is used in forensic labs. It is used to determine paternity. It is used to identify dead bodies, and it is also used to understand biological evolution. These are the advantage of DNA fingerprinting. So now we have studied the DNA, we have studied the nucleic acid, we have studied the proteins. Let's understand the difference between proteins and nucleic acid, because a lot of students get confused here. See, this is my protein. So this protein is made of series of amino acid, and this is my nucleic acid. This nucleic acid is made of nucleotides. This is the nucleotide series which makes nucleic acids, and this is amino acids which make proteins. Please note their difference. Nucleic acids actually creates protein. How? The nucleic acid use ribosomes to create, right? So protein exists as an enzyme and they perform various functions in the body. In fact, these proteins create organs, these proteins create whole body. Nucleic acid is nothing but they are used to store information. This nucleic acid creates protein, and these protein then creates lungs or body organs, and then whole body. That is the flow. Nucleic acid, chain of nucleotides. It uses ribosomes uses nucleic acid to create proteins. Proteins creates lungs, organs, and then from organs you get body. So they are all linked together. But just understand the nucleic acid is nothing but long chain of amino acids. Proteins are nothing but long chains of nucleotides. Let's take some numerical. The first is why vitamin C cannot be stored in our body. Pretty easy. See, vitamin C is water soluble. Since it is water soluble, it dissolves in water easily and can be easily excreted by urine, right? So thus it cannot be stored in a body. The next question is, what products would be formed when a nucleotide from DNA containing thymine is hydrolyzed? See, thymine will give what? Thymine will give thymine beta D2 deoxyribose. That is one phosphoric acid also. correct? So it will give you thymine beta and beta 2 deoxyribose and phosphoric acid. It will give you these three. Correct? This is my base, this is my sugar, and this phosphoric acid. As told, nucleotides consist of sugar, base, and phosphoric acid. The next is, when RNA is hydrolyzed, there is no relationship among the quantities of different bases obtained. What this fact suggests about the structure of RNA? See, RNA, if you see, is no one structure, right? So this is DNA which produce small packets of RNA, right? And these all RNA are different. So if you see, they are saying that they are hydrolyzing this RNA and they are getting different output. In one RNA they are getting output 1, another output 2. That means all the RNA are different. They have different sequence. DNA is same for a given person. DNA is same, but DNA creates RNA, right? And RNA are different because different RNA is created to create different kinds of protein, because RNA, when it is used by a ribosome, creates protein, right? So we need different kind of proteins, so we need different kind of RNA, obviously, correct, in the body. So DNA creates different kind of RNA. DNA is long chain, so small chain replicates, create one RNA. Small chain replicates, create one RNA. So all the RNA are different. Thank you. Visit examfear.com to watch more videos, attempt free online tests, get free study materials, find tutors and mentors, and much more. Thanks once again.